For as long as humans have been aware of themselves, they have noticed that children look like their parents. And of course, they don't look exactly like their parents. They're certainly not clones. And there's a variability between different kids in the same family and how much each of them look like their parents. This inheritance has been something that people have been curious about. And by 19th century, so this is when Charles Darwin is around, the prevailing idea in Western Europe was that it was the mixing of blood during conception that caused maternal and paternal traits to mix up, and the child then therefore is some sort of a hybrid of the two parents. This is where we get phrases like pure blood and royal blood. Charles Darwin really struggled with this concept because remember that in his proposal of evolution by natural selection, individuals in a population have diversity among them. And some of those individuals survive and some do not. The ones who survive are the ones who have a trait that allow them to get more resources or survive against predators or disease in whatever way. Something about their trait allowed them to survive better, reproduce, and pass the same traits that allowed them to survive to the next generation. But how did those traits get passed exactly, like physically? How did that information from parent to offspring get moved around? Darwin had a hypothesis, and it has not been supported by any science. It was rejected pretty early on. This would be a frustrating mystery to Darwin for the rest of his life. And the irony of that is that he actually had the answer to his question right in his own home office. Dun dun dun. I'll tell you more about that at the end. So in this lecture, we will learn about the father of genetics, Gregor Mendel. We'll learn about his monohybrid crosses, test crosses, and dihybrid crosses. We'll see how the basis of inheritance is actually in chromosomes and not blood. And then talk about some traits that are more complex, and we call them extensions to Mendel because they follow the same concept as Mendel's laws, but there's more complexity built on top of them. We'll look at X-linked traits and some other human traits that are of interest. Gregor Mendel was born, actually Johannes Mendel, in a German-speaking family, but on the border of Moravia and Silesia, which is modern-day Czech Republic. I think his mom was actually of Slavic origin, so she was probably Czech. And his dad was German-speaking because that part of Europe, as most of Central and Eastern, well, I shouldn't say Eastern, Central Europe was under the Austrian Empire. So he was a son of a farmer. They had a farm for the 130 years before his birth. I think he had two or three sisters, and was just a nice guy. I like reading about him because he just has such a humbling, interesting story. Well, he decided to enter the monastery in the local town called Brno in eastern Czechia, Czech Republic. And the reason for that was, as crazy as it sounds now, monasteries were some of the most progressive education institutions of their time. So there were people who did research, philosophy, communicated with other people. And Mendel himself even said that he was choosing it because he's not going to have to worry about having food to eat and putting food on the table for his kids, because he's not going to have kids as a monk, and he's going to be part of this. I mean, monasteries are kind of like communes, right? They kind of work together and take care of each other, and he wouldn't have all the like daily worries. He wanted to also become a teacher because, yes, you can be a monk and a teacher. And his dream was crushed when he failed the oral examination part of the teaching education. And then he ended up taking more schooling at a university, which was sponsored by the monastery that he was now a part of. And at the end of that second round of education, he went through his exams to become a full credited teacher again, and unfortunately once again failed the oral examination and never did become a teacher. So it was a huge bummer, big disappointment for him. But as we will see, he ended up having a very huge positive impact on the world in another way. Since he was a farmer and had interest in, hybridizing, breeding different kind of plants on his farm where his family had lived, he was also interested in this question of how do traits get inherited by the new generation? How do parents pass on their traits to their offspring? And he decided to do some experiments just to grow a few plants in his garden in the monastery and ask some of these questions, collect data, and see what he gets. He decided to work with a humble pea plant because peas are really easy to grow in this region in Central Europe. There are many varieties that have distinct features, and you can breed several generations in one growing year. Oops, I don't know what I was thinking there. And this is the monastery that he was a member of and where he had his garden, including the pea garden. I took this photo last year, actually, or has it been 2 years now? I really like Mendel's story, and it was exciting for me to be able to visit Brno, and there are several genetics museums that are really cool, engaging, as well as a small museum inside of this monastery that honors Mendel and some of his work. And of course, I met Mendel, or the wax statue of him. Don't worry, I wasn't actually touching him, and it was totally okay. Pre-COVID, so Mendel nor I are actually wearing masks because we're unaware of all the respiratory viruses that are coming our way. So let's talk about the first thing that he worked on. Now, in retrospect, we call this set of experiments monohybrid crosses. What this means is there's only one trait that he's going to be keeping track of. Okay? I want to show you a little bit of how he did it. So this at the top here is a flower of the pea plant. Most flowers have both male and female parts all on one flower. This is not true for all flowers, but it is true for pea plants. So this part in the middle that's a little bit hard to see in this picture is the female part, and the stamen here are like little popsicles on a really thin stick with a bunch of pollen all over are the male parts. And the way that it works is the wind blows the pollen, it sticks here on the female stigma, and then the pollen actually grows a tube that travels all the way down into the carpel, which is kind of like the ovary of the flower. And then the pollen, which has a sperm cell inside of it, fertilizes the egg or eggs that are inside there. Then the petals of the flower and the anthers, the male parts, they fall off, while the style and the ovary here, they develop into fruit. So when you're eating fruit of a plant, which means many of the vegetables are fruits like tomatoes and cucumbers, you are actually eating the developed ovary that has fertilized eggs in it, right, which become seeds. Then you take each of these fertilized eggs, which have now become seeds, and you can plant them, and from that, an entire new plant will grow, right? Okay, so he was able to manipulate some of this natural process by crossing different plants with one another. So what he would do is he would cut off the anthers, the male parts, off of these purple flowers, for example. Then he would use a paintbrush to dab the pollen from a different plant, and then he would tap it onto the female part of that plant that doesn't have any pollen on it. So in this way, you can ensure that it is the sperm from the white flowered plant that fertilizes the eggs of the purple flower plant. So this was his way to control the mating instead of leaving it to wind or random pollinators like bees that may come and get pollen from all over, from all kinds of different plants, and then come and possibly fertilize one of these flowers. Okay, so this is the technique that is used. You basically artificially fertilize and select who gets to mate with whom. His goal was to study two variations of a single trait. So in today's language, we would say two variations of a single gene. And he started this process by producing or ensuring that he had true breeding pea plants for 7 different traits. Okay, what does true breeding means? It means that if you have a plant that when they are able to self-fertilize, meaning the sperm and the egg comes from the same individual, then their offspring looks exactly like the parent for that trait. So let's say here, if this purple flowered pea plant was able to fertilize itself, and all of the offspring from that fertilization, once they were planted and grew, also developed purple flowers, and then you take these second generation, you make sure that they fertilize themselves only. So each individual plant fertilizes itself, and then you get seeds. When you plant them, they only grow to produce purple flowers. You would say that this plant is true breeding for purple flowers. So we're talking about one trait that if you allow the plant to self-fertilize, it will continue to have that exact same trait as the parent over and over and over for unlimited generations. He did this for 7 different traits. Flower color, so he found purple true breeding plants. He found separately white true breeding pea plants, meaning white flowered. Ones that had flower position axial or only terminal, so flowers grow at the top of the stem versus on the sides. Seed color, yellow-green. Seed shape, round or wrinkled. Pod shape, pod color, and stem length. Okay, so he had 14, you could say, containers of seeds. And each of those containers of seeds were true breeding pea plants for a particular trait, right? True breeding for purple flower, true breeding for white flower, true breeding for yellow seed, true breeding for green seed, and so on for all of 14 of these variations of the traits. Okay. So now that you know how he was all set up, let's look at the experiment that he did, his first set of experiments. He grabbed that true breeding purple flowered pea seeds and he planted them, and they all grew to be mature plants with purple flowers. Also, in his garden, he planted the true breeding white flower pea plants, put them in the ground, watered them, watched them, and they all grew to have white flowers only. Okay, so he has mature plants that are true breeding purple flower and true breeding white flower. Then he very carefully did that whole artificial breeding technique. We say that he crossed or artificially pollinated them with one another. So made sure that purple was the mom and, so to speak, for plants, and the white was dad, or opposite. He did both ways. Then those flower petals fell off and the female part of the flower developed into a pea pod, and he saved those seeds and then he planted those seeds. So the seeds from this cross were planted in the ground. When they started growing and they started maturing, he noticed that 100% all of the offspring of that cross produced purpled flower. They were purple flowered. They weren't like light purple or purple with white dots or some sort of an in-between color, diluted or pale. They were the exact purple that the purple parent looked. So this was the interesting question. Like, look, I just crossed these two different flower-colored plants, but somehow I'm not seeing any white flowers in this offspring generation. He called that first offspring generation of the cross F1 generation. It stood for first filial generation. The word filial is not used very much, but it means of or due from a son or daughter. Okay, so it is related. He didn't just make up some random word. Okay, so mystery, like, you crossed a purple parent with a white parent and all the offspring are 100% purple. What he did next is very, very important. He allowed all of these F1 generation plants to self-pollinate. So they were, this one individual plant is both the male and the female, or the mom and the dad. Then when he allowed them to self-pollinate, he collected the seeds from that fertilization and planted those seeds and waited for them to grow. And they grew into mature plants, which started to flower. What he saw in the result, which he called the F2 generation, is that there were now mostly purple flowered plants, but also some white flowered plants. This again was really interesting and kind of shocking, because imagine that for all of these plants, Both parents, mom and dad, were purple looking. They were purple flowered plants. And yet the offspring, some of them are white flowered plants. How is that possible? Where did the whiteness for this flower color come from? So the offspring resulting from this self-fertilization of F1 plants is what we call the F2 generation. And although it was hidden in the first filial generation, there is a trait that whiteness reappeared in the F2 individuals. And when he counted how many there were, which there were thousands of plants that he planted, it was about a 3-to-1 ratio of purple to white flowered pea plants in this F2 generation. And you need to remember this ratio of the F2 generation of a monohybrid cross is 3-to-1. He called, or rather, actually I can't remember if he called it or if we're calling it now. He called the trait that was the only trait of this setup in the F1 generation. So there was only purpleness. He called that trait dominant. And the one that happened to be hidden in the F1 generation, he called that one recessive. Okay, so when you breed two varieties of some trait that are both true breeding, the offspring of that cross will only look like one parent. Whichever parent it happens to look like, that trait, in this case, this purple coloredness, is dominant. And the one that happens to be hidden in that F1 generation is by definition the recessive variant of the trait. Okay, he did this many, many times over several years. And he didn't only use his true breeding seeds for flower color, he also did the same type of experiment and collected data from all these other pairings of variants of traits. And notice how many individual plants he was counting. I mean, you have 1,000 plants here for this flower color, and then when we're talking about the seed color, it's many thousands of plants. And in every case, the ratio of the dominant to recessive trait that pops back up in that F2 generation is 3-to-1. So there are 75% of the dominant trait that come up in this F2 generation, and 25% of all individuals in the F2 generation are white. This is all these percentages and all of this is very, very specific to this exact setup that he had. So you had to start out with two types of true breeding plants for the same trait. So the trait is flower color, one variant is purple, one is white, and they're both true breeding for these variants. You artificially cross-fertilize these two true breeding plants, and you have the F1 generation where 100% of the offspring come out to look like only one of the parents for the trait that you're keeping track of. So he doesn't care about any of the other traits in this experiment. He's only looking at flower color. Okay. And then when he lets F1 self-fertilize or self-pollinate, the offspring, 75% of them are the same color as F1, while 25% of them are now revealing the trait of that recessive variant. So I really encourage you at this point to pause this presentation and to retell the story of Mendel's monohybrid crosses as if you were explaining it to a classmate who missed this lecture. So start with Mendel was doing plant experiments with peas. This is what he got to get started. Make sure you understand what it means when we say true breeding plants, and explain that to your imaginary classmate that you're talking to. And then tell him what he did. So here he did 2 rounds, 2 generations before he got to the result that yielded the 3-to-1 ratio. And then go ahead and come back after you've told the story. If there's any confusion, please consult your textbook or go back and see if it'd be helpful to listen to some of this again. I hope you're really solid on the story of the monohybrid cross in Mendel's pea garden up until this point, because it gets more confusing if you are not quite solid with this material. So as he began to analyze the F2 generation and why it has this 3-to-1 ratio, he was kind of limited by the science or the technology or both that he had access to at the time. But he did figure out that the ratio actually was a surface ratio, and that deeper on the inside, the ratio was actually 1 to 2 to 1. That's super mega confusing. So let me show you what that means. He discovered that out of all of the plants that he got for the F2 generation, 1/4 of them were true breeding dominant. 50% of them were not true breeding, and 25% of all the F2 generation plants were true breeding recessive plant. This is what that means. Remember that the original parent that he started with, which we called the P generation, he had a true breeding purple pea plant, which means that inside of this plant, there is only information to ever make offspring that are purple colored. That's why when you allow this plant to self-fertilize, it will produce purple plants. And if you let it self-fertilize and make sure it only is using its own pollen to fertilize its own eggs, it will always produce flowers that are purple, flowers that are purple. And remember that the other parent that he used was a true breeding white pea plant. That means that in the cells, which he did not know much about that at that point, there's no concept of DNA at this point, but he knew that whatever it was, the information for flower color in this parent was only to make white colored pea plants, because anytime you allow this plant to self-fertilize, it only produces white flowered plants indefinitely. However, when you cross-fertilize these 2 different types of true breeding pea plants, all of the offspring are not, all the offspring are purple flowering offspring. However, this generation of purple flowered peas are not true breeding, because when he let this F1 generation, after the first cross, when he let them self-fertilize, some of the offspring, remember plants can produce hundreds of offspring because of all the seeds, right? Some of the offspring were white. By definition, true breeding, all of the offspring needed to have that same exact trait that the parent had. And here you go. It's proving that not 100% of them did. And when he allowed these F2 generation plants self-fertilize to see, well, I'm going to let this guy breed with himself and see if he's true breeding, he found out that that plant was true breeding. and that the white plants were all true breeding. But about 50% of all the plants in the F2 generation, when he let them self-fertilize, they produced both colored flowers, meaning they were not true breeding. So again, out of the F2 generation, to the naked eye, the ratio was 3-1 or 75% to 25% purple flowers to white flowered plants. And remember, for this pea plants, all the flowers on one plant are the same color. But under the surface, when he did all those follow-up experiments to figure out if they're true breeding or not, he found out that all the white ones are true breeding, but out of the purple ones, a bunch are not true breeding. And the ratios for those hidden information was 1 to 2 to 1. Okay, I recommend that you pause one more time to restate out loud to yourself or someone next to you what I have just explained about the F2 generation. And what I mean when I say 3-1 is actually 1 to 2 to 1. All right, welcome back. The conclusions that he came to is that his plants definitely did not show any kind of blending the way that people talk about blood blending as if it's like paint. You know, you have yellow and blue and then you mix it together and you get green colored something. That's not what he saw. Clearly, from that very, very, very first cross that he made, when he crossed a purple flowered plant with white flowered plant, the offspring were all completely purple. There was no blending. So he concluded that there are no intermediate traits and no blending, and that each trait is intact, discrete piece of information that gets passed on. So there's information for purpleness and information for whiteness. For each pair, so remember I showed you he looked at 7 traits and each trait had 2 variants. Well, for each trait, one of the variants was dominant and one was recessive. I think the word trait here is used in a confusing manner, so I apologize. But I hope the reason for this is because we're trying not to use the super scientific terms yet, but we will come to them in just a few slides. Okay, so in the case of the flowers for the pea plants, the purple is dominant over the white. Alternative traits were expressed in the F2 generation in the ratio 3/4 dominant to 1/4 recessive. So this is 3-1 or 75% to 25%. And he eventually described a 5-element model that we have altered just a little bit with more modern language. So parents transmit discrete factors, meaning they pass on discrete pieces of information. So like a recipe, and this is called a gene in modern terms. This word did not exist in Mendel's time. Each individual, so if you're a pea plant or a human, you receive 1 copy of a gene from each parent. And this is called the law of segregation. We're going to come back to it in a minute. Not all copies of the gene are identical. This is something that I tried to tell you when I was first explaining what a chromosome was and what a gene was. And I mentioned that you can have a gene for eye color, but there can be blue eye color versus brown eye color, right? So same gene, or quote recipe, can have variations, and the variations of a gene are called alleles. So alleles are alternative form of a single gene. In the case that we've been talking about so far, the pea flower color has two alleles, the purple allele and the white allele. If you are a true breeding pea plant for the flower color, we called you true breeding, and that's what Mendel used because that's the term that kind of all the farmers used, which is where this information was relevant at the time. But now in modern genetics, we say that true breeding, meaning you have two of the same alleles for a particular gene, we say that you are homozygous for that gene. If you have two alleles that are different, then you are heterozygous for that gene. So if I can move back for a second, this guy was homozygous for the flower color, and he was homozygous dominant. This guy, this other parent that we call true breeding, is homozygous recessive for the flower color gene. Whereas this guy here of the F1 generation, he's heterozygous. Okay, so we don't say dominant or recessive because he has both alleles. He has both varieties. They're just hidden and only one is being expressed. And again, right here, the left panel of the F2 generation is homozygous dominant. Can you pause and say what these other ones are? Okay, let's check how you did. So the left one I already said is homozygous dominant, and we only know, you wouldn't know it by looking at the plant. The only way to know it is to let it self-fertilize, breed it, and then see what all those offspring look like. And then you see, oh, it only had purpleness in it. It only had information for purpleness because all the offspring are purple. So we can conclude that this was a homozygous dominant individual. Whereas both of these examples over here are heterozygous. Again, by looking at all the purple flowered plants, you wouldn't be able to tell by just looking at them which one is homozygous for the purpleness and which ones are heterozygous. The only way to know it is to do all these breeding experiments. This white plant right here is homozygous recessive. It only has information for the whiteness, and we know that the whiteness is recessive to the purple. How do we know that? Because in a situation when both of those traits are combined in a single individual, right? The seed that grew into this plant had both information about purpleness and information about white, but for whatever reason, it only made the purpleness for the flowers. And just by that, we define purple as dominant. So if there's both traits involved, but only one of them is expressed, then that trait that's expressed is called dominant over the other one. That fourth point was that alleles remain discrete and they did not blend. There was nothing light purple or purple and white stripes or blotches or polka dots or anything like that. They remained totally discrete, unblended. The presence of allele does not guarantee expression. Remember that F1 generation, they were all purple, but they did contain in them information for how to make whiteness, and that ended up popping up in the future generations, right? So just because someone looks to have a certain trait, like you look purple, it doesn't mean that there's only purpleness information inside. Right? And two other terms here, genotype is the actual alleles that an individual contains. So in the F1 generation, that first offspring generation, that set of plants had purple and whiteness inside, so we call that genotype. Genotype cannot be seen with the naked eye. Genotype can only be found by experimentation or some other invasive, deep scientific procedures. Phenotype is the actual physical appearance, so that's something that you can tell by looking at it. So phenotype is purpleness, or phenotype is the whiteness in this particular example. Okay, so when someone says, what is the phenotype of that individual, they're usually talking about like what trait can you see or measure with kind of your naked eye without looking at what's inside the cells. As promised, we're coming back to the principle of segregation. The official definition of the principle of segregation is two alleles, remember that's versions, of a gene, which is like the recipe, segregate during gamete formation and are rejoined at random, one from each parent, during fertilization. It's almost like you have to translate that sentence. So I encourage you to pause here and see if you know all the terms already. Go ahead and see if you can digest what that very dense sentence is saying. All right, I hope you did that pausing and digesting. I will show you on a diagram what this means, and it all has to do with what happens during meiosis, which is the formations of eggs and sperm learned about in the last chapter. And of course, Mendel at this point did not know anything about chromosomes or meiosis. None of that has been discovered or described yet at all. So look at this diagram, and remember here you learned in the previous chapter that you receive, let's say this is chromosome 1, that largest chromosome that I showed you in the fluorescent picture. You receive one of those from mom and you receive one of those from dad. And in the same area of both of those, you have information for how to make flower color. So let's zoom in on that. Remember that chromosome is made out of DNA double helix that is wrapped around histone proteins that are condensed on themselves. And it is the order of nucleotides in that section of DNA that creates the code for the trait. So just like a recipe or the fact that different order of letters written down on a page have a different meaning to you, right? Like A-N-D is different than D-A-N, even though it uses the same letters, right? It has a different meaning. And versus Dan. Okay, so notice how here the genetic code, remember nucleotides are made of ribose sugar, nitrogenous base, and a phosphate, and it's the nitrogenous base that is responsible for the code, while the other part of DNA nucleotides are identical, and they just form this kind of backbone of the DNA double helix, while the nucleotides nitrogenous bases are the ones that stick out. So see how here it's TATTTAGCCA, while from the other parent you inherited in that same area a code that goes GATTTAGCCA. Notice there's one difference here. Because of that one difference, and sometimes it's more than one, and normally genes are thousands of nucleotides long, so we're just using a smaller example. Notice that one nucleotide difference in the genetic code makes it so that when this code is read by your cellular machinery, it gets a message of make purpleness in the flowers, whereas this one reads make whiteness in the flower. In reality, in most things, especially with plants, if it's white, it just means it makes no pigment, whereas reading this code, it makes a pigment that reflects purple wavelengths, so it looks purple to our eyes. Okay, so one more time, the two homologous chromosomes you inherit from each parent, and you have 23 sets of homologous chromosomes that you inherit. Each pair of homologous chromosomes carries the same genes on them, but the types of genes or the versions of them are different. So we would say that this is the P flower color gene, and these two different codes are two alleles for the flower color, or two variations for the flower color. So alleles are versions of the same gene. And this individual we're looking at, are they homozygous for flower color or heterozygous for flower color? Okay, they're heterozygous for flower color. So if you had this in your diploid cell that then went through meiosis to create an egg or a sperm, then you know that each egg or each sperm will only receive one of these. It will either receive this chromosome or it will receive this chromosome, right? So if each egg has only one of these chromosomes, and each sperm has one of these chromosomes, then during fertilization, when egg and sperm combine to create a zygote, which is finally again a diploid cell, you will receive again one chromosome from mom and one from dad, and we'll be back to where we're started here. That is taking into account a lot of stuff that we've talked about in the past, especially a chapter on meiosis. So I encourage you to pause and go over this before moving on. You're also welcome to note down any questions that you're not able to figure out after going through it on your own. In order to kind of be able to predict statistically, oh, what if I have this many of this type of plant and I breed them with a different variety of that same plant, what will happen? Something that was put together is called a Punnett square. You may have done these if you took high school biology. I remember that I did, although I didn't really understand what was going on. And they're just, it's pretty straightforward, but there are a couple of rules that when you write a uppercase letter, that represents a dominant allele. When you write a lowercase letter, it represents the recessive allele of that same gene. So in this case, they're both P for purple, even though the lowercase p actually stands for white. Okay, so they're both P's, they're both the same letter, and you can pick whatever letter you want. But they're both the same letter because they're both representing the same gene, and the gene is the flower color gene. They're slightly different, uppercase versus lowercase, because they are representing the slight differences, which is the two different alleles. Okay, so true breeding white flowered plant is the homozygous recessive plant, and that's little p, little p. Why does it have two letters? Because the plant itself is diploid. Just like most cells in humans are diploid, they have two sets of chromosomes, one from mom, one from dad. That's why you have two alleles, and that's represented by two letters. Okay. The only cells that have one allele of each trait are gametes, eggs and sperm, because those are haploid cells. So let's look at how this works. Okay, so men on top, that's how I remember that the sperm come on top. You draw a little square and you say, hey, if I know that my male partner here is heterozygous, that means that 50% of the sperm that male makes, 50% of them will get the dominant allele, and 50% will get the other recessive allele. So one more time, when a mature adult, human or plant, go through meiosis, they begin with a diploid cell, which has 2 of each allele. When that individual makes sperm, in this case, their sperm is going to be haploid. It's only going to have one set of chromosomes, so it can only have one version of each gene. Therefore, some of them, by that, remember how meiosis kind of shuffles things around until every one of the 4 gametes at the end of meiosis gets one of each chromosome. So that means 50% of the sperm will have the dominant allele, 50% will have the recessive allele. If the female, we're looking now on the side, if the female was true breeding, or in other words, homozygous, this would look, that's not on this page, sorry. In this case, the female is also heterozygous, and therefore 50% of her eggs will have the dominant allele, 50% will have the recessive allele. So this is actually crossing that F1 generation with itself. Remember that the F1 generation were the purple flowered plants that actually had both information from the purple flower parent and from the white flower parent. So all of those individuals were heterozygous. Therefore, when they themselves produced sperm, they had half of each kind. And when those individuals produced eggs, they had 50% with the purple, 50% with the white. And then you just fill in the little table, which there are 4 spots here. It's like if this sperm that has the little p happens to fertilize this egg that has little p, then the offspring will have 2 white alleles. They'll be homozygous for the recessive allele. So you draw it all in until you have this final. And this is your completed Punnett square. What that means is that when you cross one heterozygous individual with another heterozygous individual, 25% will be homozygous dominant, 25% will be homozygous recessive, and then 50% will be heterozygous. So they'll have one of each allele. And that's exactly what happened when right here at the bottom, the F1 generation was allowed to breed with itself, with the F1 generation, heterozygous with heterozygous. This is exactly what Mendel saw. Now, moving into the past one step, one generation, when you had true breeding white crossed with true breeding purple, what you actually received is that all 100% of the offspring turned out to be heterozygous. They all looked purple, which we would say they had purple phenotype, but their genotype, what was actually in their genes, was one of each allele. Okay, so here's another summary of what happened. I encourage you to take a pause and go over that last section on your own, again, explaining it out loud to your imaginary friend and trying to figure out any of the confusion that may have come a long way by reading the book or going back over the previous parts of the video. And then when you're ready to move on, we'll talk about the test cross. A test cross is like an experimental technique in order to figure out the genotype of something. Because what are the 2 potential genotypes for individuals that have a dominant phenotype? Okay, so what that means, individual that has a dominant phenotype would be you grow peas, You don't know what you're doing, but the pea plants that grew are purpled flowered. And you're wondering, is this particular plant, and I'm touching a plant, this particular plant that looks completely purple on its flowers, I cannot tell if it's a true breeding purple plant, meaning dominant homozygous plant for purple flower color, or is this a heterozygous purple flower plant, meaning that it actually has information in it or an allele for white colored. Okay. Test cross is to help you figure out the answer to that question. If an individual looks dominant phenotype, they can be either homozygous dominant or they can be heterozygous for that particular trait or for that particular gene. Test cross is a way to distinguish between those two. Okay, so cross used to determine the genotype of an individual with a dominant phenotype. And the way you do that is you take your unknown dominant looking individual and you cross them with a homozygous recessive individual. And the reason why you can do that easily is because homozygous recessive individuals, they only have one phenotype. They all look the recessive type. In our previous example, all of the white flowered pea plants, they were true breeding for white color, meaning they were homozygous recessive. They did not have any other versions of information for the flower color. They only had the whiteness. So we take our unknown individual that looks dominant. We know that it has at least one dominant allele, but we don't know if this other one is dominant or recessive, and we cross them with a known homozygous recessive. And depending what you get, you can tell whether that individual that had the dominant phenotype is homozygous or heterozygous. This is what they would look like if in the end all of the offspring from that test cross are purple, or they all look like the dominant individual, then it means that the original mystery individual was homozygous for the trait that you were seeing. If the result of your test cross yielded individuals that looked both phenotypes, that had both of the phenotypes, then it must mean that the mystery individual that looked dominant was actually a heterozygous individual. Please pause the video and look through the Punnett squares on this page in this example and see if that makes sense. So if I were to give you a result of a Punnett square on an exam from a test cross, would you be able to tell me if the mystery individual who had the dominant appearance, if they were homozygous or heterozygous?